So as Liz said, today I'm going to talk about PRIMO micropatterning for a variety of different workflows. And so the development and optimization and data collection of this has really been a collaborative effort between myself, Joseph Kim, who's a graduate student with Liz, and Jay, who you all have hopefully met by now, also in the Wright Lab and MSET Center. So first I'll talk about what micropatterning is and when it is useful. For the purpose of this talk, I'm going to refer to micropatterning basically as a method to direct cell growth in specific areas on a grid. It's a much more versatile technique than this. You can do many more things with micropatterning, but I don't have time to talk about all of them. As far as when it's useful, if you're growing cells on grids, it will be useful for you. I'll go into a little more detail why in the future, but there is some optimization. So the question is really not, will it help you, but do you need it? Is that optimization worth the benefit you'll get? And that really depends on your cells and your project. So I'll go over the protocol and workflow in more detail later, but just sort of a brief illustrated version. So you have your TEM grid, and the first thing you do is you coat it with stuff so that no cells will grow on it. Once your grid is coated, so it's PEG, there's some chemistry involved, and you project a UV pattern onto your grid using a fluorescent microscope and the PRIMO micropatterning system. And you can design these shapes to be whatever you want. And then the UV light reacts with a catalyst to remove PEG specifically in those areas. So now the idea is that your cells will only adhere to these grids where you've projected your pattern using the PRIMO system. Like I said, I'll just give a few examples of this. So we've done micropatterning for projects related to viruses. Of course, it's very popular and useful for fib milling. And Joe in the lab is also using it to direct neuronal cell growth on cryo-EM grids. So before we go into micropatterning, I just want to cover some basic considerations for growing cells on grids, starting with the least controversial. The grid must not be toxic. So copper will leach ions into your media or buffer as the cells are growing on it and will be toxic to your cells. So gold, of course, is the most common substitute. It's inert, it has good thermal properties, but you can now order molybdenum grids. They're a little bit more special order. I have not used them. The benefit over gold would be that it is a stiffer material, so should make handling better, potentially give you a flatter grid. On top of that mesh, you need some kind of substrate. And what substrate you choose will sort of be decided by what your downstream process is. So of course, everyone knows carbon. Cells grow on carbon. It's cheap, it's available. Recently, silicon oxide grids have taken off. It's a more rigid substrate than carbon. It's a little more expensive, but some cells prefer to grow and spread out better on these grids. So it might be worth that extra cost. And finally, people are using gold foil and fib milling on ultrafoils now. So if you're not going to mill, you probably want to avoid ultrafoils because the electron beam is not going to get through the foil when you're collecting your tilt series. But if you're milling, you're removing any substrate beneath your lamella, so then you can get away with less ideal substrates. Along those same lines, you need to think about how many holes you want in your substrate. If you're looking at released virions or something you're not going to mill, you probably want enough holes that your target of interest will occasionally end up over a hole where you can collect a tomogram. So we frequently use R22s or similar grids in the lab, but cells don't like growing on holes. It turns out they really like to be supported. And so that's where you might choose a grid such as the R1220s that have 20 microns of carbon in between 1.2 micron holes. And again, if you're going to fib mill, you're removing that carbon anyway, So that's not a concern from the data collection standpoint. And then finally, so the cells must remain unobstructed at high tilts and low milling angles. And so we recommend 200 mesh for everything. You can get away with 300. You might even be able to get like 1 tomogram from a 400, but I would start at 200 personally always. So this is kind of our generic whole cell workflow. So we're generally working with carbon grids, and we evaporate a little bit of extra carbon on top of the quantifoil just to provide some rigidity and support for the cells. During these workflows, there's a lot of handling of the grids. So anything you can do to help add support is great. Glow discharge them as normal. We soak them in culture media overnight. This allows the proteins in the FBS or media to adhere to the grid. We add the cells, and then, maybe we infect them with virus, maybe we label them. So there could be steps in between here, and then ultimately we plunge freeze them. And so one of the first things you'll notice if you start growing cells on grids and plunge freezing them is that they don't distribute evenly. So I know these pictures are probably pretty small for the audience, but so up here in the top right corner, you can see there's several squares that have these little dark spots that are cells. And so this to me looks pretty good. I have about 1 cell per square. I'm pretty happy with this. If I'm looking for virus, there's some space next to the cells. But then I have this whole area down here at the bottom where there's no cells. This grid on the left was grown in the exact same tissue culture dish under the exact same conditions. It also has empty areas here on the right, but over here on the left, you can see grid squares that are completely packed with cells. So in order to get good squares with good cells, you're probably going to have to freeze a whole cassette or more, and then screen them, and then load your very best ones. So what you don't see in the TEM images is what's on top of the grid bars. And when you go into SEM, it becomes very clear that cells love grid bars. And again, this is because they don't like holes so much. So the grid bar is a nice solid support. So here's just 3 different examples to show that it's not just me. Other people are seeing this. You do have some cells on the mesh, but really they're basically lining up along the grid bars. So one thing to get around this is just seed more cells, and eventually some will go onto the mesh, but then you run into the issue in the previous image of having too many cells per grid square. You also do see clumps of cells and empty squares. So ideally, your cells would be evenly distributed across the grid. And then I'll go a little bit further and say it's not enough to not have your cells on the grid bar because we're tilting. So here on the left, you see like 2 cells. You might think this is great. I'm going to get tomograms from both of these. However, when you tilt to the high tilt angles, you can see this cell that was like nestled in the corner is now blocked from the grid bar. So you're not going to get a full tomogram from that position. So this is why you want 200 mesh grids and why you really want your cells in the very middle. And the same issue occurs for FibSim. So this is a ion beam image of a cell at the milling position. And so you can see we're going to try and mill this cell here. There are some grid bars, but there's probably enough room for me to squeeze in my lamella and some trenches above and below it, and eventually collect a tomogram. But here in the foreground, you see 2 cells that are just on a grid bar. That's not going to work. But then you see this other cell here, which is technically in the square, but I don't have room behind it to mill through. So I can't mill through this grid bar here. And the same story over here. I have this cell that's nicely in the square, but it's just too close to the grid bar. I'm not going to be able to mill an area behind this cell to collect the tomogram. And so this is where micropatterning comes in. So I guess sort of the top 2 things that we are looking at for most projects is that we're trying to position the cells in the center of the squares and regulate cell density across the grid. I've found that like having an even density across the grid not only helps with data collection, but also vitrification and optimizing freezing parameters, because your thicker cells will freeze differently than the empty areas of your grid. I'll touch on this a little bit, this third point, that if you have these patterns on the grid and your cells are well positioned, you can actually use that as sort of rough fiducials for correlative studies. And then in addition to just positioning the cells, you can actually use the pattern to modify and affect your cells and potentially study the biological changes that come with forcing them into a particular shape. So there's several different micropatterning technologies. So, our lab and center, and I think in general, the field is sort of all migrating to this PRIMO micropatterning system. So one of the main benefits is that it's contactless. So as you know, EM grids are very delicate. So this uses light projection. So you're not like stamping a pattern onto your grid and risking breaking the carbon foil. It's compatible with a wide range of substrates, glass, carbon, silicone, gold, and they have a number of actual different chemistries. So if you had something weird, they might even be able to come up with that. You just design your patterns in Inkscape or Adobe Illustrator, and it can literally be any shape you want, and it can do lines down to less than 2 microns thickness very precisely. And again, it's highly versatile because you're not manufacturing stamps or you're like printing out patterns in PDMS. If you decide during your experiment that you want to modify your pattern a little bit, you just go into Illustrator, make a few changes, and then your next grid can have a different pattern. And there's even some tools within the software to do basic transformations like scaling your pattern. And so choosing your pattern is probably one of the 2 most important choices that you will make. It needs to be large enough for your cell to expand. And if you don't want to perturb your cell, you probably want it to be the approximate physiological shape. So HeLa cells kind of like spread out like little triangles. So we often use these sort of star or triangle shapes. But some of the airway epithelial cells we use tend to grow out very long. So we'll use long lines that allow them to sort of maintain their normal shape within the pattern. So another thing you need to think about is that you want your target of interest near the center of the square. So this top pattern, we wanted to look at released virions. So that's why we were trying to put the cells near the grid bar with the hope that they would release virus into the center. And then, our virus would be here. We didn't so much care that the cell itself was right next to the grid bar, but this would probably be a terrible pattern for fib milling. And then a final thing is that you need to take into account that when you load your grid into the FibSim or the TEM, you're probably not going to know what orientation your pattern was. So if your pattern is all in the same direction, then you need to be prepared for your cells to all be in the same orientation relative to the ion beam or relative to the tilt axis, which is probably not something you want. So that's why we generally, if we have a directional pattern, we try to rotate it to make sure that when we load the grid, at least some of the cells will be in the orientation that we want them to be in. And just some extra considerations, as I said earlier, the pattern will impact the cell shape and it will impact the cell cytoskeleton. So micropatterning is great, but it's not free of artifacts. So you are manipulating your sample. So this is something to be aware of. If you're interested in looking at actin networks, you probably want to be a little more careful with micropatterning than if you're interested in looking at ribosomes. Another thing is that you can be creative. So here they wanted to look at cell-cell contacts. So they actually designed their patterns on the grid bars with the hope that two cells would adhere to the bars, and then the cell-cell contacts would occur in the center of the grid squares afterwards. So again, with the PRIMO system, it's highly versatile. Anything you can think of, you can basically design. So I just want to go over a little bit of how this workflow works. So again, the first step is applying this anti-fouling layer, and it's actually 3 layers. So we do this, we glow discharge our grids and treat them as normal, and then we just set them carbon side up or top side up on a glass cover slip and add about 10 microliters of polyL lysine to each grid and incubate it for 30 minutes to an hour. And so this can also go overnight. We then add a second layer of this PEG SVA. So this will react with the polyL lysine on your grid, and this is what coats it in PEG. And so at that point, cells will not adhere to the grid. In practice, they will, but they will adhere less well. And then the final reagent, which what really makes the PRIMO system work, is this PLPP gel, which is a photocatalyst that when it's exposed to this UV light, will break the bond between the polyL lysine and the PEG SVA and release that from your grid. So before we add the PLPP gel, we transfer each grid to its own cover slip. We add this little PDMS stencil, which holds the grid to the cover slip. And this is important because we're actually going to pattern the grid facing the objective, and it's an inverted microscope. So when you're patterning, the grid is sort of hanging under the glass slide, held in place by this stencil. Once you add the gel will actually dry. And this is the only stage in the process. You want your grid to dry. And the sooner you can pattern after the gel dries, the better. But we have patterned as far as like 2 hours out after adding the gel. If you have a bunch of grids, sometimes that happens and it still patterns well. So this is, not exactly what's happening. I borrowed this figure, but this is similar to what's happening. So you can imagine, so you have your grid here, it's glow discharged, it's negatively charged, polyL lysine is sitting on top, and then you have the PEG linked to that, and we'll pretend these little water molecules are the PLPP gel. So you expose this with UV light, and anywhere that the light hits, the PEG is gone, leaving you roughly polyL lysine. And so again, so this is all done with the grid facing the objective. So it's hanging upside down off your glass slide. And the PRIMO system is attached to the back of your microscope. And it's basically just a miniature UV movie projector. So it comes with this software that you can design patterns. The projector size is such that you can pattern about 4 grid squares at once without worrying about stitching or other artifacts, although it does have tools to stitch patterns together if you want to do larger patterns. It takes about 5 seconds or less to do 1 square or to do 1 pattern of 4 grid squares. So the actual laser on time for an entire grid square is less than 2 minutes. And so just a little bit of how this works. And if anyone wants to stop by tomorrow to see the practical, you can see it in more detail. Because this is mounted onto your microscope, you have a bright field image of your grid. You can import your pattern as a TIFF from Illustrator or Inkscape or whatever. And then you can lay out that pattern on top of your grid and sort of see live within the squares, where that pattern is going to be. Just pointing out some of these 2 panels, you can repeat patterns, you can rotate patterns, and you can also adjust the dose of the laser. So something that we have found very useful is that we break the grid up into multiple patterns. And so the reason we do this is that focus is very important. So if the grid is out of focus, your pattern will be blurry. And it turns out carbon grids, especially after you've handled them and glow discharged them and added polyl-lysine and PEG-SVA, are not so flat. So by dividing the patterns into separate regions, we can focus on this, top left region, pattern it, move the stage, change the focal plane to match this top right region, pattern it, move the stage, etc. This is not so much of a concern for the silicone grids. Like I said, they have a more rigid mesh. Sometimes you can get away with those just patterning the whole grid at once. So it really takes the longest amount of time here is like orienting your pattern with the grid bars. So the whole process of like patterning a grid is about 20 minutes, and 15 of that is like getting the grid on the microscope and rotating your pattern to match the grid bars. And like I said, the actual patterning is very, very fast. So once you have your patterned grid, you need to add the extracellular matrix protein for most cells. If your cells really don't care, like let's say they're HeLa cells, you can probably just add them on the polyl-lysine that's left behind and they will grow. But most cells will benefit from adding an additional extracellular matrix protein. And this is really the second main step that I think you need to optimize. And so most cell lines have literature about what good matrix proteins are, but both the protein and concentration are very important. And additionally, we always add a fluorescently tagged protein. And so this is how I've generated some of the images I already showed you. And so this is what allows you to see actually like how well your patterning worked. So common proteins are fibrinogen, fibronectin, collagen, laminin. Again, if your cells just grow anywhere, then like you may not need to add additional ECM, but then you may not need to micropattern either. And so once you have all that optimized, you have like one choice left, and that's how you seed the cells. And so this, even though you have the pattern, if you add too many cells, they will grow on the PEG. And so you still have to be a little bit careful about how you add your cells. And so the most careful method is this monitored method. And so essentially what you're going to do, you're just going to put your grid in a dry tissue culture dish and add a tiny droplet with a low concentration of cells. So maybe like 100 cells in the droplet, and you're going to incubate that for 5 to 10 minutes. How long depends on what your cell time is. You're going to look at that on your tissue culture microscope, and you're going to see how many positions are occupied, and maybe you add another drop, or maybe all the positions are occupied and you're done, and you're going to flood this with media and let it sit for the rest of the night. If you really need, one cell per pattern, and cells not patterned areas, this is a little bit painful, but this is what it takes. It's the most controlled, and it will get you the most consistency. But we also have good success with just seeding cells normally. So you add, your full cell suspension and media, and you add your grid to the dish, and you just let it incubate overnight. This is fast. For virus infection, it also gives you a total number of cells in the dish, so you can calculate an MOI. If you use this first method, it's really hard to know, like, I think MOI is really not so meaningful. But this does give you potential less consistent cell seeding. The other benefit here, if, as I'll mention later when I show some of Joe's work, if your cells are very fragile or sensitive, they may not be able to sit around for like the 2 to 3 hours that this could take. and you may not want to like keep pipetting them. So then you'll also go with this one-step method. So this is just a quick diagram from Alveole's website to go through the protocol once more. So you're going to have this anti-fouling layer. You're going to design and project your pattern onto the grid. It's going to remove the anti-fouling layer in specific places. You add your ECM, and then if all goes well, your cells grow along the pattern. So this is actually from a pattern grid that we had made with the line pattern, and you can see the cells are nicely growing across it. So, there's a lot of optimization, but even once you have it optimized, there is a time cost to this. So it takes about 5 to 6 hours to prepare 8 grids. The nice thing is that a few of these steps can be overnight, so you don't have to spend all this 5 to 6 hours in the same day. And the grids can be stored in PBS, I know up to 2 weeks. I don't know if Joe has gone longer. If you want to know, I can ask him, or he'll be around later, I think. And you can also store them once you add the gel for a little bit if you put them in the dark. So this again, just, I've told you what it is. I just want to show you that it works. So this is patterns. So this is the fluorescent ECM on the grid. And we have just a bright field image of HeLa cells pre-plunge freezing. And then, if you look in the TEM, you see the same thing as you would expect. The HeLa cells are sort of matching this sort of star pattern I've added and growing along the grid square. And so one of our concerns with freezing was like, are these cells going to be bunched up? Or are they going to spread out? Because if your cells end up being balled up, then you're not going to get good vitrification. So if you look at confocal microscopy of this HeLa cell on a pattern grid, it's about 8 to 10 microns tall, which is the same that we see for these HeLa cells on an unpatterned grid. And that's in the nucleus, but you do have a significant portion of the cell that's maybe less than 2 microns thick. The other thing is that the cells need to be alive. And so this is just a quick stain of like live dead stain. And so it's both showing that we do have cells in the center of squares, but also that like most of them are in fact still alive. So we're going to running out of time. So quickly go through just different examples of how we've used this. So one of our challenges with our virus studies is that depending on the virus or mutant we're looking at, a very small percentage of our cells might be infected, and we don't want that small percentage of cells to be on the grid bar. So fortunately in our RSV system, which you've had some introduction to from Jay and Junlong the past 2 days, infected cells can be read. And so that allows us to localize them using CLEM. So in this case, we've imaged the grid pre-freezing. And so all of our infected cells are in red. And so this is on a patterned grid. The pattern is shown on the left. And then we can correlate that with the TEM map. And so one of the benefits of sort of how we use PRIMO to our assistance here is that we left the center of the square unpatterned, because as you know, the center of the grid square has this little unique shape. And so by preventing cells from growing over that, we could clearly and quickly identify where the center of the grid was in both the TEM and light image, and what the orientation was in both the TEM and light image. So this rough correlation happened like very quickly. And this is just another example of another TEM map. We have less infected cells. And again, so you can see that not all the cells are on the pattern. There's cells not on the pattern. By no means is micropatterning perfect. I'd view it as a way to get more cells in the center of the square. This is just some square maps, just showing you kind of the diversity of what we see of cells along these patterns. some, still like this grid bar a lot. And so just zooming in on kind of one cell here, and then just quickly play through this tomogram. And so because the cells are positioned well, we can find more events on the cell periphery than we're able to. I don't know if that's going to replay. Let's see. No, sorry, I can't play it twice. But I have this still image. So here we have like 3 filamentous RSV virions coming off the edge of the cell. And again, with the micropatterning, we're able to find more events on the cell periphery because we can position the cell periphery in the center of the square where we want it. And so, before I showed you these images of sort of cells, unpatterned cells in SEM. And so now I'll show you the corresponding panels for patterned cells on SEM. And so now I hope you can see that all 3 of these grids have cells very nicely positioned in the center of these grid squares. So you have all kinds of choices for where to set up your lamella. So this may look familiar to some of you. So this was actually one of the grids that Sergey was working with on Monday. So this is patterned HeLa cells using our little triangle pattern. And you can see that not only are the cells, going to the center of the grid squares, but they are expanding to within these patterns and growing to match the shape of the patterns that we've provided. And so this, again, can both be good, but if it's not something you want, be aware that you are constraining your cell's growth. And so finally, I'm going to show some work by Joseph Kim in the lab on neurons. So he wanted to look at primary Drosophila neurons, and there are a lot of challenges, and we weren't sure that micropatterning was going to work. And he spent a lot of time and got it to work quite well. So one of these cells are very limited and fragile. So he's digesting them from larvae, and they can't just hang around in lab all day. He only gets flies so often. And so, he really had to work hard to optimize the ECM and one-step seeding. The neurons need to grow, more than normal cells. And we weren't sure how that was going to work on a micropatterned grid, but it turns out that that is okay, that you can allow cells to continue to grow on these grids and they will remain healthy. And again, he's isolating these from live cells or live larvae. So he can't wait around, isolate cells, pattern for 5 hours, and then add the cells to the grid. And so this is where being able to store micropatterned grids allowed him to make those in advance, save them, and then use them right when he had his cells ready to go. So I told him to send me bad pictures of unpatterned grids, but I think these are actually some of his better ones. So you can see cell bodies, in the grid square, and they do have some neurite growth. But they are near the grid bars. But there are some potentially collectible areas here, and just another panel over here on the same thing. So you can make this work without micropatterning, although I think an awful lot of work and optimization also went into these images. But you really see a striking difference with the patterning. So when he does these patterns, he's looking at curved and straight. He's seeing the cell body adhere at the end of the pattern, and you can really trace and monitor the neurite growth along these patterns. And you now have a significant amount of this neurite extension, throughout your grid square, where you can, either use montage tomography to collect a large view or collect a number of tomograms across this length. And so this is just another example where he's used an X pattern here. So you have cell bodies in the middle, neurite growth extending out. And this is a tomographic slice. showing that everything that you expect to be there is there. You know, we have microtubules, we have mitochondria, everything's well vitrified. So the micropatterning seems to be supporting like normal cell growth and you know, is not inhibiting freezing or any downstream processes. So in summary, you know, micropatterning is a way to increase throughput of your whole cell cryo-ET studies. It's very complementary to CLIB and FibSim. It's very versatile, you know, any grid type, any sample. You will have to optimize per sample what your ECM and patterns are. And then again, you can go beyond just positioning cells. There's a whole lot you can do. If you go to Alveole's website or, you know, look in the literature, I'll have a slide of references here at the end. You can explore some of those additional possibilities. And so that I'll just like to thank everyone in the Wright Lab, particularly Joe and Jay, who really helped out and did a lot of work on these projects. Laurent from Alveole, Serge from Nanoscale Labs, you know, everyone in our cryo-EM centers here. And of course, you know, Mickey Woods and Tom Coombs keep our instruments running. I wouldn't have any images without them. And, you know, we've had a lot of support from the thermo applications team as well. So just like to thank them. And with that, I will take any questions.
Questions?
Thank you for that talk. It was quite interesting. I am curious, in alternative to micropatterning, you know, are there alternatives such, you know, like in general for cell manipulation? I know light or ultrasound or maybe even like digital droplet PCR, for instance. Are there other technologies that exist, maybe not catered towards the cryo DM audience, but ways to manipulate the cells? Because it seems, the reason I ask is because it seems like this requires a lot of tweaking and may be off-putting for some people to try it. I'm just curious if that's considered.
I am not aware of other technologies that exist. Obviously, there are, you know, like micro manipulations capable of manipulating single cells. You know, one thing that's coming to mind, you know, we have our Vitrojet, which is sort of printing cells onto grids. And right now it's just ignoring grid bars. So it's printing like a circular pattern, but, you know, it would just be a matter of programming if you wanted that to like only deposit, you know, in the center of grid squares. So I'm not aware that that exists, but I think that kind of technology could be developed. And yeah, it would be interesting to see if anyone's working on that or what the future is there.
Yeah, I mean, the reason I ask is because, especially if your goal is really only focused on centering the cells in the center between the grid bars, that seems like a technical challenge that could be overcome with kind of micro manipulation. Potentially it's faster. I'm not going to downplay the technical, you know, engineering challenge to also do that with a TEM grid. But I don't know, but potentially an application space that could be explored.
Yeah, I think that would be an interesting possibility.
I had two questions. The first was, when you're not using the PRIMO micropatterning, I was wondering why you glow discharge before incubation overnight. I'm just wondering if the hydrophilicity would wear off by the morning. And then the second question was, I was curious how much practically contamination is an issue, or like, so I'm guessing you can't like UV sterilize these cells after you did the PRIMO, otherwise that would obliterate your pattern.
Yeah, so the first question, sorry, was, oh, why glow discharge? So we do want the proteins in the FBS and media to adhere to the grid. So I think the glow discharging helps with that. And also just getting the grid into the media. if the grid is hydrophobic, it'll want to float and flip over. And so glow discharging, yeah, it's just lets you soak it more easily. You know, if you were able to soak the grid in the media, by overnight, it would be hydrophilic just from having soaked. But the glow discharge, I guess, is really a convenience at that point. And contamination. So for eukaryotic cells, you know, we have the standard antibiotic antifungals in our media. You know, we're careful with all of this, but we don't do it in a hood up to adding the ECM. And we haven't seen contamination. You know, if you had the neurons or something where you're doing a longer cell incubation, I would be more careful. After patterning, you can rinse the grids in ethanol. And so after you do the patterning step, you know, you can then take them into the hood, rinse them in 75 or 70% ethanol or soak them, and then wash them in PBS and do the rest of the steps in the hood. So I think that would be the best way to be sterile versus the balance of doing everything in a hood. I mean, obviously you could get a microscope in a hood, but if I was really concerned about it, yeah, I would rinse in ethanol after patterning and keep everything in the hood after that step.
Thanks.
I thought I'd add to that contamination concern. I've incorporated ethanol washing after micropatterning to all of my neuron preps now because that is the exact thing that I'm worried about. And it's worked fine.
I had a question about the PRIMO. So do you contract use of for the PRIMO for BSL-2 samples?
Yes. Our PRIMO is in our BSL-2 lab, so we can do BSL-2 samples on.
And like you can contract that out. So if like I wanted to send some samples over, that would be possible.
Yes.
Oh, nice.
I mean, the other thing is that, well, we've tried this a little bit, but we should try it more. Yeah, because you can store these grids, we should be able to make grids and ship them. So like, you know, if it's easier for us to make the grids and then you to add the cells in your lab, that should be possible. We should be able to send you like patterned grids.
And then just like one more question. So because you've done a lot of work with the HeLas and you know like they're very unhappy in the holes. And so have you seen like one type of material that they prefer best that they can grow on?
So HeLas mostly, as long as it's continuous, so I think they're sometimes happy on R2x2s, you know, it depends on the cell, but they definitely, the R1220s, like you see less membrane blebbing and things that suggest that the cell is stressed. So I'd say the more continuous surface, the better. HeLas tend to do okay on carbon, but some cells don't even really like the 1,2,20 carbon, and the silicon dioxide is yet another step at making those cells grow better.
Thank you.
Hey, that's a really interesting talk. I was curious if you've explored different patterns that kind of like optimize the amount of like the edge of the cell if you want to image without milling. For example, like budding virus or something. Is there a pattern that you found is best for that application?
We have not tried that. I don't know if that was in Julia's paper, if they tried that. I thought I had it here. Yeah, I mean, so maybe something like an X or something like this might give you a little more. Yeah, you almost need these inverted curves if you want to optimize surface areas. So I have not tried that, but there's certainly, like I said, because it's just you draw it in Illustrator, anything you can imagine, you can pattern.
Cool. Thank you.
So I do have to say we're a little over time. If you want to ask your questions, it just means our coffee break will be shorter. You want to, what do you want? You want to ask? Okay. You're second.
Okay, thanks, Brian. Very interesting talk. I just have two questions. The first is about the choice of grade. I wonder, do we have to choose the grade with continuous carbon film, or just common grade will be okay?
So I have not actually tried growing cells on continuous carbon film, purely under the assumption that it will break.
Okay.
I think like growing the cells and the handling, like the 2 nanometer film will break. But I haven't actually tried it. You know, graphene would be a similar thing that could potentially be tried that I haven't. But then again, there's concerns like, is it supportive enough that the cells won't break it? So I have not tried it. I'm not aware of anyone that has, but I think it would break, but it would be interesting if someone tried.
Okay. And the second question is, as we all know, the grid is pretty fragile. So I want let's say if I have prepared maybe 8 grids, so how many grids will survive all the process?
Survive the micropatterning process?
Yeah.
I'd say all of them generally. I mean, maybe when you're first starting out, I'd prepare a few extra, but you know, now if I make 8 grids, they'll all make it through to the end.
Okay.
It's just a few extra handling steps. So yeah, once you're comfortable handling grids, it's pretty straightforward.
So I have, I guess, 1-2 questions. So when you have the pattern, right, the cells will only go where they, you allow them to go. If you overseed, right, like say 10 times than you should have, where would the cells go, the extra cells?
So the cells will go on the peg. I mean, it depends on the cell, like how much they will, but yeah, so actually in this image, you can see like this top row has this X pattern, but like, you know, this square here, like it's grown in between the X's. So definitely the cells will prefer to adhere to the ECM, but like they will land on the peg and grow over the peg if you overseed. Probably on the grid bar, but I haven't really looked that closely.
Right. So just thinking, have you tried like overseeded just so at the end result, you still get like a similar, good patterning because they were just only allowed to grow in your pattern area. In that case, then you don't have to kind of tune your density much.
So in my experience, if you overseed it, you will get cells outside of the pattern. So it will just be too crowded. And the other problem is that the cells are much smaller when they're first adhering. So you might get 3 or 4 cells adhering within the pattern, and then they can't expand because they're all competing for that same space.
Right. Another question is, did you try much finer patterning? Like you say, you can go up to 2 microns, right? It's
actually like 1.4. I don't know. Alveoli always gets mad at me when I say under 2 microns. They're like, oh, it's 1.4. No, but yeah. So I have not tried less than 2. I don't know what Joe's lowest is.
So I definitely have patterned at the resolution limit, technically at 2 microns. The thing that I noticed is actually the limiting factor starts to become the cells just refuse to get onto the pattern if you're not careful enough. So I've gone back to thicker patterns as a result.